I'm Bruce Seligmann. I'm the co-founder and chief scientific officer of BioSpyder technology. I'm going to be talking about spatial transcriptomics. Now, that is a new technology, new methodology with several platforms, either just recently commercially available or in development. But I think spatial transcriptomics means different things to different people. And so I'm going to try to address some of those differences as well as some of the considerations in selecting a platform to pursue. So one of the aspects of spatial profiling in general is that it's particularly important in that people can use fixed tissue, and you don't have to be a pathologist or a molecular biologist in many cases to carry out assays in which you can profile cells, glands, or histologic structures of interest within the context of their microenvironment. And that's what's meant by this term spatial transcriptomics, or as I will be talking about it as spatial profiling most of the time. And one such method that I'll talk about is the one that we've developed, which is called Tempo-Seq. So that's in development at this point. It's not a launched commercial assay, but it's a gene expression assay that can use fixed tissue as the sample type. It doesn't, again, require expertise in molecular biology. And an attractive aspect of it is that the data analysis is largely automated. So it also doesn't require the support of a bioinformatics expert as well. And this is carried out through a package that's called Tempo-Seq R. But the point about this technology, the focus of it is to enable investigators to get spatial expression data from their samples, from fixed tissue that's directly stained, either with H&E staining or with antibody staining. So what are some of the factors that go into making the decision of whether spatial profiling is necessary for someone's project? And what are the considerations in terms of picking a platform to carry that study out? And the first is, well, what's to be gained with spatial profiling versus profiling a bulk heterogeneous area of tissue? And I will address that. Second is, what is the resolution? Are you interested in going all the way down to a single cell or subcellular areas, or is the interest to look at normal glands versus, let's say, cancer glands or any type of gland without getting cross-contaminated profiles from surrounding stromal tissue? So that's certainly a very important aspect in terms of picking a methodology and also ultimately in the cost and throughput. And is there a need for histologic specificity? So among other things then is to gain that histologic specificity where you're only going to profile one specific histology without cross-contamination. Is the methodology, does it require you to use a reference section that's stained and then the sample is actually taken off of a second unstained section? Or can the section that you're actually working from and profiling, can that be directly stained or not? And that brings then to the four is the type of sample. Can you use fixed tissue, or do you have to use fresh frozen tissue? Because that dictates obviously what types of samples and how many samples and how complicated processing those samples can be. And what is the method of sequencing or of readout? Is it a 3' biased method? Is it targeted? And then in context of that, what is it that you want to measure? Do you want to measure just what genes are expressed? Do you want to measure splice variants or expressed SNPs or fusion genes or other mutations? Do you want to measure small non-coding RNA or long non-coding RNA as well? All those are questions. In fact, even if you want to measure histones, which are not polyadenylated, then the point is whether you would want to use a 3' biased method or a different method. And another question is then ultimately, what is the performance? What is the gene expression measurement, both the sensitivity and the spatial resolution that can be achieved. Can the platform deliver the spatial resolution that you need to measure the area that you're interested in, and can it do so with sufficient sensitivity and specificity? And then finally, of course, is the cost and the throughput. If you're only going to look at a few sections, then maybe that's not quite as important, but if you certainly are going to look at lots of sections from different patients and in lots of areas within those sections, then both cost and throughput are really critical questions to ask. So to address the first issue of, well, is a heterogeneous sample good enough? This just shows a section of prostate tissue that's actually used for TMAs, which is what those circles are left over from. And standard RNA-seq methods take a fair amount of material. The standard Tempo-Seq method takes 1 millimeter squared or less, which is shown by the green box. So what's in that 1 millimeter area? So this is a blow-up, and you can see it's still very complex. It has a lot of different glands, some of which could be normal, some of which can have cancer epithelium, surrounded by stroma cells in that mix. And of course, then there might be also inflammatory cells as well in there when you get down to the single cell level. So nonetheless, you can get a lot of useful information off of a bulk tissue. So this shows a study that we've carried out using colorectal cancer, where we've looked at cancer and adjacent normal using 1 to 2 millimeter squared areas of tissue. And if you do that, you can see from the methodology viewpoint, if you look at normal tissue between different patients, each patient is color-coded. The normal tissue is in the square boxes over on the left. They're all fairly tightly grouped, which shows what is the repeatability between patients of the assay, because one assumes that normal tissue should be about the same between individual patients. Whereas on the right, you see the cancer profiles, and there is clustering. And so one of the questions is, well, we know that for colorectal cancer, there is a 700 gene, approximately 700 gene signature described that's used to subgroup patients into clinically relevant subgroups. And so if one looks at this data without adjusting it and just uses that microarray signature, it turns out that you can take the cancer tissue off from the Tempo-Seq data, and you can assign it to either the CMS subgroups or the CRIS subgroups. And we have one patient that we could not assign, and one patient that we could only assign to CMS, but there was no adjustment made for differences between the Tempo-Seq platform algorithms that you might want to optimize to versus the microarray. So you definitely can get very useful information off of bulk samples. But if you go back to the prostate example and look at that tissue section, and you have a 2 millimeter squared area, and then you go down even smaller than that into what's a 540 micron squared area, you see here you have a gland surrounded by stroma. The blue arrows indicate where the gland is, the orange is the stroma, and you have other glands there. And so would you be able to get the information you need if you had, let's say, a 130 micron diameter area, which is circled here in red, just looking at the gland without any stroma cross-contamination? So that's the type of question to ask. And in this particular method, with Tempo-Seq, it actually leaves a visual mark of where you profiled from. And so you can see that there's no contamination of the surrounding stroma with that gland. So one can go on and look at a lot of different types of tissues just to get an appreciation of what one is talking about. Is a single gland good enough, or do you need to go down to single cell resolution? And so this is looking at gastric pits, and you can see if there's a blow up on the right. So you can see if you want to look just at the gland, you can use a series of areas that you profile and pool together, which is shown on the left. But if you want to get to a single cell, now you're down to a 10 by 30 micron area that one would want to profile. So the methodology has to be able to achieve that to go to single cell, whereas 30 micron diameter multiple pickings certainly would work fine if you wanted to look at the whole gland. And you can go on to other tissues. This is lymph node, starting really at the more gross scale. The marker here is for 1,000 microns in lymph node. If you want to look at cervical epithelium, glands, or other types of tissues, you're down to 200 micron type resolution for a single gland within that type of tissue. So you need to be a little bit more precise, but you're not at the single cell level. What about mammary ducts and other types of bronchiole and other types of glands? You're at that 100 micron to 200 micron level of needing to have that type of spatial resolution. Primary oocytes, and now you're down to the 70-50 micron level. And this is the gastric pits is what I started off with in this little series of slides. So really, it depends on what the tissue is that you're looking at and what the question that you are asking is as to what methodology to look toward to provide that. But another consideration is whether you can use directly stained tissue or have to use a reference sample. On the left is directly stained, again, prostate. Means that you can manually go in and pick the area that you want and profile it. Whereas if you're using a reference section, then that would be the left sample, and you're getting the sample off the right sample, which is a second serial section, you now have to have fairly complex image analysis software built into the system so that it can register the reference sample appropriately so you hit the right area on the sample that you're pulling your sample from for profiling. And this slide, I see that the animation's messed up a little bit, so we'll rapidly go through there. The point is that, for instance, if you were going to look at a gland, it may be a fairly large gland that's surrounded by stroma. If you look in the lower right corner, that means your registration, however, has to be good enough that that edge of the area that you're profiling doesn't overlap into the stroma. So the spatial resolution has to be much higher than the area that you may want to be profiling. And it's going to be down at the 10 to 5 micron area. And the problem with that, and using a reference sample, is that that gland in a 5 micron or a 10 micron section is going to move its edges in the next serial section. It's not going to be identical. It's not perfect round or perfect cylinder. It's, it's contorted. And so there's no guarantee that gland will look like that in the next serial section. And so those edges are going to change, and you can get cross-contamination then if you're using a reference sample down at this level of 100 microns or 130 microns or less. It becomes much more difficult to use a reference sample. So I wanted to cover a couple of the technologies, and so what I've done here is gone to publications so I can talk specifically about that publication. And so this is Spatial Transcriptomics, which was really the first of the methods out there. And it's a method where you basically print an array on a slide of capture molecules, put frozen tissue down on top of that, release the RNA it's captured, and then you profile. So it's a grid. And in this publication, each spots of the grid was 100 microns in diameter, and then there on center was 200 microns. It's my understanding that they're going to resolutions down to 50 microns as this is now being developed more completely. And it uses a reference section, which is shown here on the left, which is stained, and then can be profiled by the pathologist as to what the areas are, and then that is then correlated then to the grid of profiles that one gets off the assay. It's a 3' biased method is carried out in this publication using fresh frozen tissue, and it was 10 microns thick of melanoma lymph node. A note about thickness. So if you're at 10 microns, that's actually your limit of resolution because you always have depth as well. So if you are trying to get less than 10 microns, then you have to cut thinner sections as well because you have to think of this in 3D and not just in terms of the surface area. So again, if you're going down to single cell resolution, you have to get to think about the thickness of the sample as well. So spatial transcriptomics approach is a grid, and as you can see in this image of the 100 micron, it's heterogeneous. It's not histologically specific because the grid doesn't necessarily line up with small focal histology. It certainly can cover larger focal histology well, and that's what's shown in the annotation up above. Is fairly large areas of lymph node histology. The second method, which is the Tempo-Seq method that's being developed, can use directly stained H&E or antibody-stained tissue, and I've already shown the fact that you can profile from a single gland. But if that gland happens to be elongated, then one can take smaller areas but pool them all together to cover glands which are elongated rather than circular. And this uses the Tempo-Seq method, which is unbiased, but it's a targeted sequencing method, so it measures specifically what you have in that assay in terms of sequences, but it can measure exon junctions, splice variants, in other words, and can measure gene fusions, SNPs, and other sorts of molecules that are not polyadenylated. So, and then the last method is the GeoMix Digital Spatial Profiling that NanoString has launched recently. And so this is a publication that has just come out. And when it comes to gene expression, the spatial resolution is 300 microns. And so even though the instrument can go down lower than that in terms of spatial resolution, in terms of to get data, because of the sensitivity assay that's useful, you need a fairly large area to do that. In this case, they were measuring 88 genes, tiling across them, so they're looking at multiple sequences for each gene, 770 sequences using the N-counter assay, 928 for sequencing. And in this case, you can directly stain using RNA-scope to look at RNA levels and then pick those areas. And you can do protein, but that would be in a serial section, not the same section. And finally, FISSEO, which is a single molecule fluorescence in situ sequencing, is a method where you can look at subcellular gene expression. And so this is in situ sequencing, so it is slower than the other methods in terms of having to actually sequence the image rather than collecting sample from the tissue and then sequencing offline. And right now, this method is providing about 200 reads per cell. I believe they're working to get this up to around 8,000 reads per cell. So it looks at a few genes, but it's not restricted into what genes. So you can look at any genes, but it's a smaller number because of the depth of reads that one can get out of the system. So basically then, the method, whether it's 3' based or targeted, is important. And if it is targeted, can it measure the whole transcriptome or not, is important to know as well. So I wanted to talk a little bit about the Tempo-Seq assay platform. And so this is a methodology which can be used in FACS-sorted cells, can look at single cells, and cytosine-type preparations or fixed tissue. And it has single-cell sensitivity. And so I want to talk about this because one of the questions is the method one's looking at to have true single-cell sensitivity? Obviously, FISSEO does, and how does one go about assessing that? As I said before, it can use fixed tissue that's H&E stained, and it can either use simple lysates from that sample or an in situ protocol. But either way, there's no extraction of RNA or reverse transcription required, so it's a very robust, simple method, and it has the automated analysis. So this just shows a question to ask when looking at a technology or an experiment to do, which is to ask if I have a bulk cell sample and I compare that to a single cell from that bulk. So how does that correlation look? In this case, you see that the bulk cells correlate very highly to the single cell by a fluorescence activated cell sorting, and you can see low expressed genes at the single cell level that are in the bulk sample as well. So you can basically see every gene, but there is stochastic expression. Some genes are not expressed at the instant that these cells were fixed for assay. Minutes later, those genes may be expressed and other genes may be off. You can take a cytospin and ask then whether the focal method, the focal spatial profiling method, also has single cell sensitivity. And so this is taking a single cell using a 30 micron diameter aspirate of that single cell. And you can see that it gives the same single cell results as being able to see low expressed genes that you can see from the bulk sample. So this is a test that you can do with any method out there to ask what is its performance, and am I limited to only looking at moderate to high expressed genes, or can I measure every gene that's measurable from bulk? This happened to be MCF7 cells, and it shows true then single cell sensitivity. The other question to ask is, what is the repeatability between single cells? And so this is, again, looking at the FACS assay because it's easy, but it's the in situ assay, looking at two single cells. And again, you see stochastic expression, but you see very good correlation between those two single cells quantitatively. So this is not looking at differential expression, this is looking at counts. So it's direct number of molecules in each single cell being counted. This is looking at a whole transcriptome, 20,000 genes, and using a depth of reads of about 500,000 counts per sample. But that's something you can dial in. So the stochastic expression, which is circled in red here, can be caused by the method itself, would be a technical issue, and it can also be biological, inherent in gene expression from single cells. And so there's a component of both, and that's something that one needs to be aware of is that stochasticity due to low expression, or we're not measuring every gene that's measurable or should be measurable from those cells. So I wanted to sort of move very quickly and sort of towards the end here to, well, what is utility of spatial profiling? And so this table, I think, is a good presentation summary of what we have observed and which I think is highly significant. And that's namely that if you take an area and you scrape it, you look at the column that's on the left, in each case it's called scrape. Those are the levels of gene expression from that scraped tissue that contains primarily cancer epithelium, but also normal epithelium and stroma and immune cells as well. And then if you go in and you do spatial profiling using the same sort of resolution as before, 130 micron diameter areas, you see in this cancer line, these top genes here on the left, upper left, are expressed in cancer. A subset are expressed in PIN. PIN is a precancerous histology. But none of these are expressed in normal. Whereas with the bulk tissue, they're all there. So you get a biomarker specificity as a result of spatial profiling. And you can go through to look at the PIN in normal. And so now you have a set of genes that are expressed in normal, not in cancer, and then a subset are in PIN. Adhesion molecules are expressed in all three. Stromal markers, however, are not expressed in any of them, showing that you do have no cross-contamination from stroma. Housekeepers, other types of genes on the upper right are expressed in all three, as you would expect. But noteworthy is, in the lower right, is immune markers and cytokines are only expressed in cancer and only in a subset of those expressed in PIN, nothing in the normal tissue. So basically then, with using the focal analysis, it identifies that you can identify what are histologically specific sets of genes. And this has led us to hypothesize that the tissue microenvironment exerts phenotypic specificity that's lost outside of the tissue setting. That means if you dissociate them, you won't see that. So you have to look within the context of the tissue, and it could be the neighboring cells exerting this, it could be factors, extracellular factors exerting the specificity. But if so, then histologic-specific spatial resolution is going to provide a biomarker specificity that is never seen in heterogeneous tissue assays. And it may be possible then to obtain insights into the mechanism of disease and its progression, and to identify novel therapeutic targets, which are not possible to identify using heterogeneous bulk FFPE samples. And it also may be from this increased biomarker specificity able to get clear stratification of patients using focal profiling rather than heterogeneous bulk tissue, and therefore better prognosis, better diagnosis of disease. So basically, those are things which we can talk about generally, but there's another aspect for Tempo-Seq is that more recently people have been using that to do high-quality dose-response data. And so that means using focal profiling, it's possible that one could get high-quality dose-response expression studies carried out at the level of the spatial profiling to understand how a target histology within a tissue is affected by a compound or drug, just not only for translational studies, but also to determine drug efficacy and side effects for drug discovery as well. So whatever you can do with a bulk, you potentially can do at the level of spatial profiling. So, of course, we have the questions, can we validate the hypothesis, and is it true beyond cancer? So Tempo-Seq, there's a fair number of publications. What I would like to sort of draw your attention to are the two at the bottom. The fixed tissue assay has been published, not the spatial profiling assay, but the bulk assay. But in particular as well, the use to provide dose response data and complex analysis across the whole transcriptome or across a set of 2,000 or more genes has also just recently been published, and I think does represent a whole new avenue of investigation and opportunity, both within bulk samples and vitro studies, and then at the spatial resolution level as well. So I would like to thank you for listening to the presentation, and I'd particularly like to thank the people who contributed to the Tempo-Seq data from BioSpyder and also consulting pathologist Raymond Nagel. Thank you.